Good morning, good afternoon, and good evening, everyone, and welcome to this webinar titled Achieving Consensus When Everyone is an Expert, but No One Agrees. This webinar has been sponsored by the Partnership for Health Analytic Research, so a big thanks to them for helping to make this webinar possible. We're very fortunate to have three speakers joining us today. First up, we have Dr. Jinsi Paulos, Executive Medical Director and Team Lead for Non-Malignant Hematology at the U.S. Oncology Medical at Novartis Pharmaceuticals Corporation. Jinsi and her team have conducted several Delphi panels in sickle cell disease. We also have Dr. George Joseph, who is Vice President of Health Economics and Outcomes Research at Bausch Health. George has experience using the modified Delphi method to develop clinical guidelines in hematology. And lastly, I'm happy to introduce Dr. Michael Broder, your moderator for today and the president of the Partnership for Health Analytic Research. Michael has conducted dozens of expert panels on a wide variety of topics. Hi, Michael. Please take it away whenever you're ready.
Hi, great. Thank you. Hi, and welcome, everybody. I appreciate you being here, and I hope we make this a useful and informative session. It's mostly going to be a panel discussion, but at the beginning, I'm going to just right now do a little introduction of myself and my company, and then we'll introduce the other presenters. We'll do, in the midst of our webinar, we're going to do a few audience polls just to get a sense of where you all are in terms of your knowledge level, and so we can aim the conversation at the right level. So I'm, as Sydney said, I'm a physician and researcher, and I've been working with the modified Delphi panel method for a long time. My background is that I started my career as an academic OBGYN, and I worked at UCLA and the RAND Corporation as a researcher. And RAND is where this modified UCLA RAND process began, and so I got some experience using it back then and have continued to use it throughout our now almost 19-year lifespan of PHAR. And PHAR is a health economics and outcomes research consultancy, and really what we focus on is problems where you need to bring together clinical experience, some HEOR methodology, and really highly trained people to make it work. And we're a relatively small group. We have 15 full-time researchers based mostly in Southern California, and we concentrate our work on real-world data. So that's secondary data analysis and primary data collection and analysis, and then some other things, including, as I mentioned, this modified Delphi panel method. And I found it a very useful method for a wide variety of things. We're going to talk with Jinsi and George about some of the ways they've used the method, but it's been used and can be used in a lot of other settings as well. That's just our business has been, we've managed to keep a high level of academic rigor, and as a result have quite a few publications over the years of PHAR. And with that, I think I'd like to introduce the other panelists, or have them introduce themselves, really. So welcome, Jinsi and George. Thanks for being here. So, maybe you can go ahead and introduce yourself. Jinsi first.
Hi, hello everyone. Thank you, Michael, for the introduction. My name is Jinsi Paulos. I am the Executive Medical Director and the Team Lead for the Non-Malignant Hematology franchise in the US for Novartis. Had many various experiences working with PHAR in sickle cell disease with various panels that we've incorporated. As part of the Team Lead for Non-Malignant Hematology, we always try to identify gaps in the medical space in any disease state that we're looking at and try to address these gaps in the landscape that's provided. And we've utilized the consensus model for a lot of these gaps that have been identified. Thanks, Michael.
Great, thank you. And George?
Thank you for having me on, Michael. So I'm George Joseph. I'm the Vice President for Health Economics and Outcomes Research at Bausch Health. We cover hepatology, GI, and dermatology as our main areas. And the application that we'll be talking about today in the seminar will be primarily on how you use a Delphi panel to sort of conceptualize actually a health economics problem and trying to figure out, what is the best way for us to sort of conceptualize the disease and then sort of quantify the disease in a health economic model. So I'm very glad that we were part of the Delphi panel for sickle cell disease that I got to work with Jinsi on.
Thank you. Maybe the first thing we can start with is sort of how you got interested in doing the panel. And George, you just alluded to that before we started, so maybe you can talk about that a little bit. What was your goal and why you thought this method would work?
Well, honestly, I didn't even know the method would work, but the way it started off was just like Michael said. I also did my doctoral training at UCLA, and so I was exposed to the Delphi panel. It was part of the health services research curriculum where they teach you research methods, but it was just.
Oh, George, we've lost your audio.
In our organization. That was the first application. So there was a bit of curiosity saying, yeah, I know it's a Delphi panel. I know academically what it means, but how does it actually come into being? So that was the best part about actually being experiencing the Delphi panel. Why we did it. Without going into the specifics, so Jinsey and I were sort of working on sickle cell disease for an asset that was going to be approved by the FDA in, say, about a year or two from the time that we were actually looking into it. Jinsey and I had the pleasure of working with many medical experts. Each of them was an expert in their own field. Some of them specialized in pediatric sickle cell disease. Some of them specialized in adults with sickle cell disease. And the reason why we were talking to them was because we wanted to sort of conceptualize the patient journey and sort of understand the severity of the patient. And every time we spoke to an individual, they were like, yep, this is definitely my patient journey, but this is not something what Dr. X or Dr. Y would agree with because they treat a different kind of a patient. What about this particular adverse event or this particular condition? Oh, yeah, that's important for me, but if you're over 40, that's not important. So what we were figuring out was everybody was treating the disease, but everybody had a different viewpoint depending on their own clinical exposure and clinical experience. And we wanted to sort of get a handle on that.
Yeah, that's really interesting and useful to hear that background. Maybe right now is a good time for me to. I have a few slides on just sort of the basics of what is expert panel, and so maybe I'll do that because I see, like I said, about half the people really had never done one before. So let me go through some kind of high level things like this. So what do we mean by expert panels? First of all, I'll say that I use the term to mean something fairly specific, but there isn't a commonly accepted definition of what it is. But when we use it, what I mean is a structured consensus method that brings together multidisciplinary experts with the goal of getting consensus on topics where there isn't yet strong empirical evidence. If there's 6 randomized trials on what to do, then you don't usually need expert consensus because it's obvious. And I don't use it to mean an ad board or an unstructured discussion among experts or a literature review, even if it was done by experts. So that's sort of what I mean when I say it. There are a lot of different expert panel methods that people talk about. Another term people use other than expert panel is structured consensus process or structured expert elicitation. And these are different types of methods that I'm showing here. And maybe I'll speak to just a couple of them. The nominal group process really is the concept of a nominal group is what it means is a group that is a group in name only. And that means you gather experts together and you allow them to share their opinion, but you don't really let them have an argument or an interactive discussion. So people are sharing their opinion. It's kind of a one-way street flow of information. And actually the RAND UCLA modified Delphi process uses part of that, and I'll talk about that in a minute. And then there are other processes, many of you are familiar with the NCCN guidelines, and they use a structured consensus process. I mean, they don't use really a formal process like the modified Delphi method, but what they do is they set standards in advance for what the level of agreement is before they can put something in the guidelines. So they need 85% of the panel to agree on it, and if they don't, they change and they do some iteration. So they have a structured process. BOGSAT is a term that I only learned recently. I'm surprised that I hadn't heard it before, but it stands for a bunch of old guys sitting around and talking. And I think this was the traditional expert panel method. I wouldn't consider it a good method, and it's not something that we use. The Delphi technique I'll mention because many people have heard about that. The Delphi is something that was developed also at the Rand Corporation in the 1950s as a way to gather expert input and help them achieve consensus. And the way it works is you develop a questionnaire and send it out to your experts, and they answer it, and they send back the results. You collect the results and aggregate them, and then share back with the experts where each of them lies in terms of the group median or mean. So are they in the range or outside of it, or where's their answer compared to others? And they're given an opportunity to change their answer. And sometimes in some iterations of it, not only to change their answer, but offer the reason why they gave the answer, or why they continue to give an answer that's outside the range. And in a typical Delphi, you'll do multiple rounds. The panelists never meet together. And you either say in advance how many rounds you want to do, or you say when we reach a certain level of consensus, we stop. And that technique was used for many years successfully. What the folks at Rand and UCLA thought was that it would be good to have an opportunity for the experts to talk amongst themselves. And so therefore the modification of that process. And this little cartoon just shows kind of at a high level what are the elements that we use when doing a modified Delphi panel. So you gather your experts together, and then, you get this list of experts, and then you develop a literature review and share that with them. The idea being that even though they're experts, as George said, some are expert in one thing and some are expert in another. And then you make this rating form. The rating form is really a very detailed questionnaire. And usually the questions are about specific types of patients. Each question, it's laid out in a grid, and I have a picture I can show a little later, but the questions are laid out in a grid, and each cell in that grid represents one patient scenario. And there are hundreds of those for a typical panel. And this is circulated to all the panelists, like in the Delphi, without them meeting the first time. And they answer those questions usually on some 1 through 9 or 1 through 5 rating scale. We collect those responses, aggregate them, and score them. And by scoring, I mean we make it easy to tell where there's agreement and disagreement by color coding them. And then you get the panel together, either in person and or over Zoom, and we could talk about some of the differences in those things later on. And then when they're together in person, either myself or another professional moderator moderates the discussion amongst them, and we focus on areas of disagreement. So we really try to get them to talk about things that where their answer was outside the range of the other answers. And we don't let them try to convince each other. It is really just the sharing of their reasoning. And so that's why I think that's an element of the nominal group process. And at the end of that conversation, which lasts a full day, often or more, they do the ratings again. Those second round ratings we take, aggregate the scores again, and from the areas of agreement, develop statements we call consensus statements, and then share those around to the panelists for them to decide if they agree. That's a typical process. It doesn't always work exactly like that, but just at a high level, so everybody is kind of on the same page. So I hope that's a little helpful background. Maybe let me ask Jinsy, when you did the first, you've done several panels, I guess. So what was your interest in, and did you consider alternatives, or what did you find different about the different panels that you did?
So, as a medical lead, right, we tend to do advisory boards. And as George mentioned, George is the one who actually introduced me to PHAR and this actual methodology aspects. And so we haven't tried any other aspects because like NCCN guidelines, obviously we can't have any, decision aspects in, right? But it is a way that we find useful to bring these like guideline aspects to the forefront where there may not be any guidelines, right? And as George mentioned, like severity of sickle cell disease, there's no guidelines. No one's ever actually addressed it. And so it's a way to bring gaps in the landscape out to, in front, as well as, provide guidance, especially to physicians who may not be experts per se in that disease state.
Mm-hmm. Mm-hmm. Yeah. And actually, George, did you have something you want to add, or?
Actually, yeah. So I don't know if Jinsi, you remember. So when we introduced the concept of sickle cell disease severity, many of our medical experts had a sort of a visceral reaction to it. They're like, sickle cell is a disease. I think we're doing a great disservice to our patients by even calling them less severe, more severe, and all of that. And that itself triggered it. So even though they had no common basis or consensus on what defines a patient, because they all treat different patients, one thing that there was definitely uniform consensus on was the fact that we would be doing a great disservice by calling a sickle cell patient less severe or more severe. So that is why we had to very carefully choose even the terminologies to even describe what the disease is as the patient progresses through it.
Yeah,
Yes, we had to use a grading system
right.
similar to
Okay.
like cardiac. So it was very
Yeah.
interesting.
One of the things about this panel that the panels in general that I enjoy and that I think speaks to really what you're talking about is that in the development of this rating form that goes around, our process is that we talk to all the panelists multiple times. So it's not like I'm sitting there developing the questionnaire without their input. So each panelist we try to have at least 2 conversations with where we're sharing kind of here's what we're thinking, these are the questions, and during that process, I think all of the panelists in the experience that you're talking about became more and more comfortable with the goal of what we were doing and that it would be useful to do. But
Yeah,
since
but it doesn't work out that way, right? Sometimes.
no, right. Clinical practice guidelines is the highest, and that's not a surprise. Actually, in the 80s, when this method was developed, it really was developed with an eye toward creating clinical practice guidelines. That is really what the idea was, or measuring the appropriate use of procedures or interventions. Those 2 things are pretty interrelated. If you say something's inappropriate, it means it's a bad idea to do it, and appropriate, sort of guideline level care. So yeah, that's interesting. So people have had experience really across the board. So even though the development of the process was for sort of appropriateness of procedures, my first experience was in the early 90s related to hysterectomy appropriateness. But it's been used for a wide variety of things. And I think it's a technique that really what it does is it helps a conversation happen. So you have these experts that all know these things, and for them to be together and just have a free-ranging conversation, there's no telling where it's going to go. But by moderating it and by creating the structure beforehand, you really help them develop a common language for how to talk about what they disagree about. So we have an example, and maybe I'll show it now because I think it might make sense to show this. So this is what a typical rating form would look. This is one bit of a rating form. It might be too small to see, but I'll give you the general idea. This is a sickle cell document that we were talking about. And so on the left side in the row headings are different patient characteristics such that each row represents one kind of patient. So this table is like, I don't know the number, but it's maybe 24 different types of patients. And then the columns are different questions. And we use those questions to help the panelists think about which patient type is the most severe. And then in the final column, we asked them how severe is this patient, and we gave them a rating, a way to answer that on a scale. And then here's a completed rating form. Again, just one piece of it. A typical Delphi panel rating form for us has between a couple hundred and maybe 800 of these cells to fill out. So panelists really do a lot of work beforehand in getting this done. And this document shows the color coding. Yellow means disagreement. And so in that conversation that we're having, we're trying to focus them on areas of disagreement. And that's another thing that I think really helps make it not take, we do spend a lot of time, but we try not to spend a lot of time on things where there is agreement. So and then just finally, one more image I'll show you is what the output looks like. So we take the results of this. So in this case, these dark blue cells represent, the most severe cases, and the light blue are where people think that's the least severe. And we turned this kind of a completed rating form into a flow chart that allows somebody to, a clinician, to answer a series of questions and decide, is this patient, as George and Jinsey both said, they didn't want to say severity, so they used this class 3, class 2, class 1 language, and that's what we ended up with. So maybe let me ask, what about the virtual versus, I was just talking about how we get the conversation going. And Jinsey, maybe talk to me about your experience in terms of virtual versus in-person panel. How has that worked?
Yeah, so I've done quite a few of these for various different projects, right? Not just severity. And just to preface everybody, mine are unblinded. So you can have a blinded consensus Delphi panel as well as an unblinded. There's differences to each, and blinded meaning that the company or whoever's sponsoring the event will not know who the participants are. They only see the final outputs. There's only one chair that's nominated, and then that chair is responsible with working with PHAR to ensure, to identify the other experts, and not allowed to listen or anything like that. The unblinded ones that I've done, where we try to limit our participation because we do not want any kind of bias from the pharmaceutical industry that we represent. And I've been able to do unblinded because they're not product specific, they're disease specific, right? We have had one that's product specific, but that was specific to an adverse event for our drug. So kind of, you knew it was from our company. So with the blinded, we identify a chair, and with the chair, we, ask the chair, who do they nominate as experts? And then we look through the list and see, okay, yes, this person makes sense and that doesn't. And we want a varied type of disciplinaries, right? Like George mentioned before, pediatrics versus adult. Depending upon what the Delphi panel is, we'll have a patient. We've had that as well, who was a patient as well as an HCP, because we want to ensure that HCPs are comfortable speaking their mind and what they would actually do. And in front of a patient, it may be varied, just to ensure that patients don't come out with a bad impression or anything like that, right? And then also like psychosocial, hematology, immunologists, whatever the aspects of the Delphi panel requires, we include those experts in as well, right? And the PHAR team does a very great job ensuring, and it's a very long process. It's not something that's just quick and easy done with, it does take 6 to 9 months. And PHAR has been really great in ensuring that if we have a milestone that we need to reach, they will help us out. But just for everyone to understand, it does take 6 to 9 months of understanding it. And it's because of what Michael said, creating these rating forms in conjunction with those panelists takes time, right? You have to you might have one question, oh, how do you do this? But then how an expert interprets that may be different amongst all the panelists. So then you have to modify those questions so that everybody's speaking the same thing, right? You want to make it sure that it's comparing oranges to oranges versus apples to oranges, right? And so that in itself takes the longest time. Then when it comes to the actual meeting, so I've done live and I've done virtual. The virtuals that we've done have actually been broken out into 2 days, 4 hours each, just because of the fatigue involved with screen times in front of the computer. The live ones have been the 8-hour ones. I find the live ones to be more beneficial just because of the fact that the HCPs are truly dedicated to their time, right? You're in front of them, they're in front of you. There's less chance of them having to be able to multitask because it is a discussion happening in front of your peers so that you're more engaged, right? The virtual ones, they're engaged. I'm not saying that they're not, and they are participating. And Michael does a very good job. Michael's been my moderator from all of my Delphi panels. He does a very good job ensuring that, especially the quiet experts, that they get called on and they're able to provide their input as well, where they're not overshadowed about somebody who's a little bit more vocal in their opinions. So Michael does a great job in that. But in the virtual setting, obviously we all know with the pandemic and everything like that, you're multitasking. You can turn off your camera. Things happen where they're supposed to dedicate some time to us, but nanny calls in sick or something like that, and they have to take care of kids, or something happens, right? So you don't get that full focus that you would really want to have, even though they're engaged. It's easier when you're seeing everybody's face and on the screen, and you could tell by facial reactions and everything like that, right? That's where live comes into play, because then you can read body language a lot easier than over Zoom. And then you can pick up on if someone's comfortable versus uncomfortable in a specific topic, or what another expert is saying. I think we faced a lot when we did the triage one, right, Michael, for sickle cell disease in the ER in New York City. We brought about a lot of experts. And in there, we also had a patient as well as who was also a physician. And you could tell from the body language of everybody when there was a clear disagreement, like everyone was like on edge, and it's just like, what are you talking about? You guys, you spend so many hours with Michael's team trying to come up with these. What do you like? And they change their mind at the last minute, like, no, we're not doing that. I'm like, okay, so you get to read a little bit more. Whereas in a virtual setting, it's a little harder, I would say.
Yeah. No, that's right. I mean, my experience so we've done probably half a dozen virtually, during the pandemic. And I was really afraid that it was going to be a disaster the first time we did it. And I was surprised at how well it worked. And people do pay attention. The disadvantage, as you said, is it's when the panelists are away in some setting in a conference room, they can look at their phones, but they're not going to disappear for an hour. That just doesn't happen. And we've seen that happen in the virtual panels a couple of times. But the advantage of the virtual panel is that, one of the things you mentioned as kind of a disadvantage is that the time it takes to do to get it all set up. And the advantage of doing it virtually is that you can do it a lot sooner because people don't need to make travel plans. In the row here at the top is a patient that has this is sickle cell disease, no end organ damage, and no chronic pain, and doesn't visit the ER unexpectedly during the year. And at the bottom, you have somebody who has severe end organ damage, is in chronic pain, and goes unexpected ED visits multiple times a year. And so each of these patients is slightly different. And by making those differences very slight, you force them to confront what actually is driving their decision. So is it the number of ED visits? Is it the fact that there's end organ damage? And what is it exactly that's doing that? And so that is like, if you've read that Thinking Fast, Thinking Slow book, it's the system 1 versus system 2 thinking. And so we're forcing them to kind of be novices again, to think about what their decision making is. And that is part of why they're able to have a conversation that isn't just an argument about, oh no, in this case I do this, and in this case I do this. They really have been forced before they show up to think about how to answer it. And really, I think the rating form, one of the main things it does is when people come together, they have thought about all of these individual aspects, like, oh, is it age? Is it lab tests? Is it some other comorbidity? What is it that's making me make this decision? And so that first round of ratings actually isn't used at all. We get their ratings, but we only use it to compare to the second round. The second round is what drives the consensus. So, okay, so all right, good. We have some time left, so maybe let's talk about a few other things. I mean, George, you talked a little bit about the sort of the process of getting it going, but maybe talk about the difference between this and an ad board and how kind of institutionally you had to what magic you had to work to get them to understand what you were trying to do.
Could we go back to the poll results so we could sort of sequentially look at that? Yeah.
I think I can do that.
I think it's like 28.
Yeah, but I think, okay, let's see if I can make it. Oh yeah, here we go. Mm-hmm. Can you see that, or let's see, push right
now. So I'm hoping that if you press view results, you can see all of them. So I'll definitely like to echo what Jinsy said. But I think for the audience, you have to sort of understand what your own organization's ability to review and approve an ad board is. So think of it as explaining this to your legal and compliance team as, is this an ad board or how is this different from an ad board? Are you able to do an ad board in 4 weeks in your organization? I mean, I haven't seen one, but I'm assuming that there are organizations which can organize ad boards in 4 weeks or maybe even 8 weeks. So yes, this does take time because there are two levels here. One is where I see the third one saying unfamiliar to organize one. Everybody's unfamiliar, including your own legal and compliance team, especially if you're in pharma. So I think the first challenge for Jinsi and I was that we had to sort of present the concept of a Delphi panel to our legal and compliance team, differentiate saying that, no, this is not an ad board. We're not just flying them into Jersey City for 8 hours, doing slide review and sending them home. There is a lot of pre-work that needs to be done. That is going to be happening. So the only part that is common with an ad board is flying them down to New Jersey, in our case, to have them sit in a room for 8 hours for an in-person Delphi panel. But there's a lot of homework, a lot of discussion that Michael and team will be having with individual medical experts even before they fly in to meet in one place. And there is a defined goal. So if legal and compliance is, okay, so what do you want to do with all of this? So we had a defined goal that we need the team to sort of come up with a consensus, or at least document what was the final consensus or final understanding of the sickle cell classification. And that documentation would become a manuscript if all the ad board medical experts believe that that is something that needs to be published. So with legal and compliance, we said, this is how it's different. Yes, it's common that they all fly in, but there is a very defined objective. There's a lot of homework, and there might be, or a very strong possibility, there might be a publication outcome that results from this particular exercise. So I think that's what we went round and round and sort of trying to explain to our legal and compliance team. Yeah. Every organization is different. So, and then of course you have to go through contracting and all of that. But the scientific part, as sort of Jinsi alluded to, was 6 to 9 months because there's a lot of homework involved. If you have 10 KOLs, that means at least 10 1-hour meetings. Can you organize 10 1-hour meetings in 1 week? Or with medical experts, that would be like 10 weeks.
Yeah. No, that's right. That's exactly. I mean, our experience, and not just with you, but I've seen that happen at other organizations too. It just requires a little bit of a champion who can argue for why this is necessary in this moment, and what is it that what is it that we're going to get out that you aren't going to get somewhere else. And
Yep.
Oh yeah, go ahead, Jinsi.
Oh, I was going to say, and after you do the first one, they know what it is. And so then it becomes a lot easier to get these kind of approvals. You just have to make sure that you are asking the right question, right? What is the data gap that you're trying to fill with this white paper? And just providing that rationale. But yeah, after our first one, George, I haven't had any issues trying to explain why we need to do a Delphi panel or what a Delphi panel is.
The other thing I see, hard to find experts, and so maybe let's talk a little bit about what that process is like and how you do that process. So Jinsi, you want to talk about that and how people were selected for the panels? Or
yeah, and as I mentioned before, right, you just need that one chair, one expert that understands what you're trying to address using the Delphi panel, right? Once you have that one chair, that chair can help you identify your other experts. You just need to tell them, okay, I'm looking for a pediatric, I'm looking for an adult, I'm looking for this, and they will help you. So don't be afraid to ask your experts to help you with that. Now, with the disease severity, we had our chair. We had some people that we wanted, but we also asked that person, can you please provide us who you think would be appropriate? Because you also want to ensure there's a varied amount of experience, right? That everybody's not the same. And they know their peers a lot better than we know their peers and how they interact with each other as well, right? So I think that that's the main point, like find your one chair and the rest will be easy, supposedly.
Yeah, that's good. I mean, I do think that technique has worked many times to start with the chair or somebody like that. We aim to usually get between 9 and 12 panelists altogether. And we also start out with some concepts about the categories that those experts have to be in. And so we don't, sometimes the you'll have an idea like, oh, I want this person and I want that person. But what we try to do is back up a little bit and say, okay, what are the categories of knowledge that you want represented here? And as part of the modified Delphi panel method, another concept is that you want people who are not just the academic clinicians who on this panel. And I've had this experience where, and so we always try to get community physicians and people not from just from the ivory tower. And sometimes that's a little bit of there's a little bit of push and pull about doing that because, from the pharma side, often you want the people that you really think of as the key opinion leaders. And that isn't, you can't just stuff the panel with those people. And I think we had this experience with one of the panels that I did with you, Jinsey, and I've had it before, where having somebody who's practicing outside of academia is super useful because they can say, well, it's going to take me 8 hours to get that test result. So there's no point in ordering it to try to make a decision. And you need to know that that's the reality. And so we do that. And you also mentioned earlier, Jinsey, about having a patient representative. And I think personally, that's really important to do. Patients often on these panels are a little reluctant to, kind of talk. You're asking these clinical questions. And of course, they can't answer, I would give this drug in that setting. But what they can do is that they can listen to the conversation and say, what I care about as a patient is that my pain is being treated quickly. Like that's the most important thing from my perspective. And so that I think is really useful to have. And so you're trying to fill in all of these different, kind of categories of expert. And that can be hard to do, but using this technique of either one expert who serves as a chair or even just somebody who's not formally a chair, but will, do these things, that's very helpful as well. So we're about a quarter to the hour. I maybe want to think there might be one or two more things we can talk about, and then I want to leave some time for questions. One of the things you mentioned, Jinsey, was blinding, and so tell me what that's about and why you did it, if you can.
So the blinded ones that we've done at Novartis were because we didn't want bias towards our specific product, right? So to ensure that there was no bias in that aspect, we did it blinded. So we wouldn't pick anybody. They wouldn't know it was our company that was sponsoring it or anything like that, just because we didn't want that bias. So it was done by class agents versus product specific. Now, my experience is only with unblinded, as I said before, because it's disease area aspects that we're trying to uncover. And then the one I did product specific, the adverse event was very product specific. And so regardless, it would have, even if we tried to do it blinded, they would have known it was us, right? So
what we found, what I've found, so when I started doing this work, none of them, we didn't even have a concept of blinding the panels. And then I think appropriately, reviewers began to comment on how can you have a panel of experts that you paid to talk about, prescribing your drug and then think that's unbiased. And so we developed this concept of blinding. Either the panelists don't know who the funder is, or sometimes, as you mentioned, the funder doesn't know who the panelists are. And it adds some complication to the process, but surprisingly not that much. The hardest part is getting contracting done. Sometimes the pharmaceutical company is a little bit uncomfortable agreeing to pay people who they don't know who they are. But we've managed to work it out, and I do think that it helps when you're trying to publish the results of these things. If you can say, if it's a product specific issue, as you kind of pointed out, Jinsey, a lot of the things that we're doing with this panel method really aren't talking about using a particular product, like the development of severity classification, and we've done other things that are like that. That's not really, it doesn't help you any more than it helps, it really helps the body of treating clinicians more than it helps you as a company. So I think those are, that's something that can be done.
There's a question about publications when they're blinded. So regardless if it's unblinded versus blinded, you as a company representative are never authors on these publications. At least I have not been, because we did not feel as a team that it was appropriate for us to be authors on a publication, especially when it talks about guideline aspects, right? We wanted to be from the experts themselves. Our only role was funding or sponsoring the guidelines. We try to take a step back in the aspects of when they're just when the rating forms are created. We don't have the individual conversations. Michael's team takes care of all of that, right? Even when the rating forms are developed, we can ask clarification questions, but we try not to interject ourselves too much because we do not want that bias, right? When you're doing consensus guidelines on treatment paradigms or whatever not, you want to be far removed because as much as you can from a pharmaceutical aspects, because you want to limit whatever bias could be from that. But all publications, our company names are never on there as authors, blinded versus unblinded. It's all from the experts.
Yeah. Yeah, and what we've done when it's blinded, what we've done is we unblind it after the first draft of a manuscript is written, because for the disclosures for the people writing the manuscript, the authors, they need to disclose who paid, and at that moment they don't know. So we write a paragraph in the methods that says, everybody was blinded until the first draft of the paper was written, and we've been pretty successful. And so the idea is really just to say that while these conversations were going on and during the process, nobody knew who was paying for it. And of course, at the end, they do know. Then you said something else that I wanted to pick up on, and I can't remember. But maybe. George, any other comments you want to make? Otherwise, maybe we'll go to the questions and I'll see what questions are, and we'll try to answer them.
I think there's some very interesting questions that are coming up. I think we should try and
Okay. All right, great. So I'm looking at these, and maybe here's how well accepted are the published results of the Delphi panel? Have you seen many examples where it really changed how physicians thought about a particular topic? Yeah, that's a really interesting question. So in my experience at RAND, what we were doing was using these panels to develop criteria for whether a procedure was appropriate or not. And those we actually did before and after studies, after the guidelines were promulgated, and they did have some effect on practice. It's not as changing practice is very difficult. So where they really had an effect is if an organization made an effort to take the guidelines and implement them. So some organizations like, a Kaiser Permanente, for example, or some other big, organizations do kind of have internal processes where they try to get guidelines used. And so in a setting like that, it makes a bigger difference than if they're just published and left there. But you have to measure it to know. I think they have an impact, but how big, I think it sort of depends. Let's see. Okay, here's another one. Probabilistic elicitation to explore subjective uncertainty around key parameters has been accredited by NICE in the UK as the preferred method for eliciting uncertain model inputs. Does the panel have experience with this, and to what extent can the RAND UCLA method be used for these purposes? Yeah, so actually,
there are two parts to it. So in the example that we gave you today, we were using sickle cell. And all the slides that you saw, the disease itself is treated differently in the US versus the UK. That's because there's a lot more patients with sickle cell in the US. So without going into too much specifics, there was a US model being built, which is what this was going to support the conceptual framework. There was also a parallel effort for an ex-US, or what you call like a NICE model that was being built. And again, if you can go back to that slide 29, the clinical parameters that US physicians weigh may not be the same as what a UK physician will weigh, because can we go to slide 29 or 28?
Yeah, I'm trying here. I think I got it now.
And so what our recommendation is, if you want to submit, and I'm using sickle cell as an example, because the disease is very different in how it's treated in the US versus UK versus in Africa. I would recommend building your own conceptual framework as to the classification of that specific disease if it's different across the framework. And that's very, very important. So a US physician is not going to really care about what they do in the UK because they have different standards, different protocols in the ER, and different metrics. So be mindful of that. So that's why we try to build two separate parallel models, one for the US using the Delphi panel, and our colleagues worked on an ex-US, let's assume it's a nice model that was being built with different set of clinicians who practice in that particular country and therefore could conceptualize the model as it was fit for them.
Yeah, that's a good point and a good answer. And I think also to the questioner, and maybe to the questioner, I would also say that most of the work that we have done has been to develop clinical guidance, although we have used this method to develop estimates of various parameters for things that have been submitted to NICE or other regulatory agencies. And a couple of years ago, the Decision Support Unit at NICE came up with a kind of a list of methods that they thought were appropriate to use when there was uncertainty around estimates. And expert elicitation is clearly one of them. They at least in my understanding of it, they took a little bit of a agnostic stance about what the best method was. But we've used this method to come up with estimates, specific estimates of cost when there's no when there isn't a clear number there, and also for quality of life when we dealt with a very rare disease and we used the method to come up with estimates of quality of life for various disease states when there was just nothing realistic in the literature. So we have done that. Okay, another one says, could you please give us a bit of insight when no consensus is actually achieved on one or two key criteria, and how do we report the non-consensus? Yeah, that's a good question. So there are always places where there isn't consensus. It's just not in the nature of these types of groups to be able to come to 100% agreement. And what we try to do is report the areas of agreement and disagreement. Usually that there's some meaning to that disagreement. So in that, what's a good example of it? We often say in certain areas, clinicians recommend, or the experts recommend, in this setting do this, and in this setting either A, B, or C. So that's kind of how we would handle the disagreement, that people there was too much spread. And we also, I'll just say a little bit more about disagreement. We usually use a kind of a technical definition of agreement that says that people can't be too far spread along the answers. So if everybody's kind of clustered in one place when they answer, then that's agreement. And if most people are clustered and they're one or two off on the edge, that's still agreement. But if there are people all over the map, that's disagreement. And there's some numerical ways of doing that. But really, we try to incorporate that into the guidance that comes out of the result. And then here's another one. We've gotten this question before. How do you deal with consensus feedback that's not in line with labeled indications? Yeah, I mean, what I would argue and have argued is that these are expert clinicians giving their opinion. They are not bound by the label. As you know, practicing physicians can prescribe anything for whatever reason they think is appropriate. And so the resulting consensus statements and publication are not a company sponsored. They're not saying those things. The company isn't saying those things. It's the clinicians saying them. So it hasn't been a problem. We've done panels where the entire question is practically outside the scope of what's the label indication, and it hasn't been a problem. I can see that, as George talked about, like kind of talking to legal and compliance folks and convincing them, and Jinsy mentioned in some ways too, and I think it's probably just a matter of helping them to see that that's what you're really doing, is you're helping to guide these clinicians to share their knowledge. It's not, the company that's saying what to do here. Let's see. Anything else? Somebody had asked, I knew there was a question about numbers, and I tried to say, I mean, we use 9 to 12 as our kind of ideal number, and that's sort of traditional. I've done them bigger. It's hard to have a conversation when it gets much bigger. I did one in for end of life care, appropriate use of end of life care. We had about 20 people. What happens is that it's really too hard to get each person to share their opinion on a particular area of disagreement. That takes too long. So you have to use this method where you get people to offer their rationale, and then you just kind of have to ask, does that match other people's rationale for why they answered? So they don't get as much opportunity to interact and talk. So I think it's not as enjoyable for the panelists. And just it's a harder slog for the moderator. In a Delphi, not the modified Delphi process, but in a Delphi, you can use a lot more people. And so that can be a method to where you can get a lot more input. So it's relatively easy to send out these surveys and aggregate the results. So 20, 30 people is reasonable for a Delphi that doesn't have an in-person component to it. So what else? Let's see. Any challenges? Do you have any? Have you had any challenges using the chair to recruit from an ethics compliance perspective? We haven't. We don't ask the chair to do the recruitment. Just to be clear on that, what we're doing is we're eliciting names and then matching those names to the areas that we still need people in. So, do you know somebody who's in community practice in the western part of the country, or do you know a radiologist who does this at another hospital, and that kind of thing? And then we talk to them. So that works out okay.
And then as long as you tell your medical and compliance group, like the recruitment is done, through, advice from your chair as well as the medical team. There usually is no questions regarding that.
Well, I see we're really at the end of our time. So I want to say thank you all for your attention. I hope it's been useful. I think we got to a lot of the questions. If you have questions that we didn't answer that you want answered, we will do our best to try to answer them offline, either with an email to you, or we're going to kind of put together answers to the questions in a document and make that available to you. You'll get an email about that. And so maybe then I'll just turn it over to Sydney.
A big thanks to the audience here for participating as well. And last but not least, we'd like to thank the sponsor, the Partnership for Health Analytic Research. In closing, we hope that you enjoyed this webinar, and we'll see you again next time.